Hello and welcome to the Spotlight on AT Research webinar brought to you by the Global AT Alliance. Now, the Alliance was established in 2020 to maximise efforts, resources and funding to find a cure for ataxia telangiectasia. Members include patient groups and charities from around the world. They passionately believe that with increased funding and a continued global and collaborative effort, effective treatments for AT will be developed sooner and the lives of those affected will be changed for the better. Now, my name's Naga Munchetti. I'm a broadcaster, journalist and a charity patron. And I'm joined in the studio for the next 2 hours, hopefully with you as well, by the Chair of Action for AT Research Advisory Committee, Dr. Mark Toms. Mark, it's good to see you again after a short while apart. What have we got on the agenda today?
Yeah, great to see you and great to be here this afternoon. So we've got a great lineup of speakers from around the world today who are going to talk us through some of their work, some of their studies in a concise and easy to understand manner. And then, of course, we'll have an opportunity to answer some of the audience's questions.
And you want to know how to answer questions, of course. So you'll have a chance to, after each presentation, via Slido and vote on various polls throughout the event. So here's a quick guide to asking questions via Slido.
We're excited that you're joining us for this digital event today, and we really want you to get involved. There are a number of ways you can do this. Use the round Ask button in the bottom right-hand corner of the page to post a question. You can do this with your real name or post anonymously. If you want to upvote a question that's already been asked, use the thumbs up button. Throughout the event, our speakers may activate polls which will appear automatically. Please do get involved with these so that our speakers know what you're thinking. We really hope you enjoy this event today, and now it's time to introduce our first speaker.
We will do that in just a moment, but I'd love to see if you can follow instructions and use Slido, of course. We'd love you to tell us where you are today, where you're joining us from, and we'll show you. We've got a wide variety of attendees. Fabulous. Argentina, South Africa, we've got loads in Europe as well. United States, the Bahamas. Greetings from Bolivia, someone has said. Greetings back, of course, and from Dimitris as well. Thank you so much for taking the time to join us today. It is really good to have so many members of the AT community from all around the world. Now it is time to learn more about some of the current AT research studies taking place at various institutions all over the globe. Sit back, enjoy, and please don't forget to use that Ask button on your screen to put your questions to us. Up first, we have Borja Saiz from the Clínica Universidad de Navarro in Pamplona and Mark Well from the Universitat Pompeu Fabra in Barcelona. Hello, Borja. Hello, Mark. Really good to have you with us. Really looking forward to this first presentation. Take it away.
Yeah, thank you so much. Naga and Mark, thank you for organizing and hosting this event. I'll be starting the presentation, and I'll give the second part from the presentation to Mark. I would like to start by thanking AT Alliance and in particular IFAD for supporting our research throughout the years, and most of all the families and all of those kids afflicted by the disease who are our true inspiration, I would say, to continue doing this work. So resetting the immune system, I should say resetting by replacing the immune system as a method to fight immune deficiency. So next slide, please. So as you all know very well, mutations in ATM, which is a gene which is implicated in DNA damage response on the correction of DNA mutations, is the leading cause of AT. Perhaps not the most characteristic, but one of the major clinical features in AT is the immune deficiency that these kids have, which is basically the inability to mount a correct immune response and therefore to fight infections the way we usually do. Immune deficiency is perhaps the main cause of admissions in hospitals and definitely one of the major causes of concerns in families, and therefore something that we are trying to tackle with this project. Next slide, please. So immune deficiency can be resolved by a bone marrow transplantation, or so-called the hematopoietic stem cell transplantation, aiming at replacing the immune system. So just briefly, the hematopoietic system and the immune system is hierarchically organized, with a stem cell, which is a very rare cell that can proliferate and differentiate into all the effector cells in the blood, including the immune system, B cell, T cells, and the neutrophils and macrophages, for instance, and give rise to all those lineages. In order to achieve a bone marrow transplantation, what we need to do is to basically eliminate whatever is endogenously in the bone marrow and then replace it with cells from a donor that is healthy. Or in the case of AT, a successful bone marrow transplantation will most likely require the ex vivo manipulation of the hematopoietic stem cells of the kids by means of editing or gene therapy, and then the reinfusion of those cells into the organism of the kids. Those cells will be able to engraft and differentiate, giving rise to an immune system which is proficient on AT and therefore will not have the primary immune deficiency that is characteristic for the disease. Next slide, please. So why is the bone marrow transplant not used these days regularly as a therapy for AT? And there are two issues that are the major hurdles on this race. One is technical, perhaps, and it's the fact that ATM is really a big gene, so it is really, really hard and very low. The efficiency of encapsulating of the genetic material on vital particles is hard, and therefore the efficiency of the correction on hematopoietic stem cells is extremely low. And the second issue that we faced is that in order to achieve a successful bone marrow transplant, you need to precondition or perform a preemptive treatment on the patients that is extremely, extremely toxic, and that hampers the use of BMT in AT. So these are the two questions that we're going to try to tackle with our project. Mark will actually talk to you about the first issue and the solutions that we are planning to implement, and I will be talking about the second, which is the conditioning regimes. So next slide, please. So conditioning is basically, as I was saying before, to eliminate the immune system of an individual, so the corrected or the healthy stem cell can engraft, proliferate, and differentiate, giving rise to all the immune system. The issue is that killing all the immune cells of an individual, but also all the other hematopoietic cells, will actually lead to low numbers of leukocytes circulating, as well as clotting factors, which may put the life of these patients at risk of infections, but also bleeding complications, etc. On top of it, the conditioning regimes that are used in the clinic are based on chemotherapy or the combination of chemotherapy with radiation, and the extreme hypersensitivity of AT patients to these agents makes it impossible to use, or the risks are too high to basically consider it on a regular basis for AT. Next slide, please. So what we have come up with is an alternative way of conditioning, which instead of ablating the whole immune system, we are trying to get rid of the stem cell, which sits at the top of that hierarchy, by using a vehicle, which is an antibody in this case, conjugated to a toxin. By this means, what we aim at is to eliminate those cells while preserving the rest of the immune cellularity. Those individuals will not have that decrease in the number of leukocytes, and therefore they will be able to fight infections while the corrected stem cells are engrafting and differentiating. Next slide, please. So our studies in preclinical models in the mouse actually suggest that a single injection, single administration of this immunotoxin in ATM deficient animals is actually extremely potent, eliminating stem cells in the bone marrow of these animals. Next slide. However, the cellularity in the lymphoid B and lymphoid T, so the adaptive immune system, as well as the myeloid cells, and even more in the progenitors that give rise to all these cells, is not affected, suggesting that while we are capable of eliminating the stem cells at the top of that hierarchy, the rest of the cellularity is intact in these settings. So now the next question, and if you go to next slide, please, is to see whether on top of eliminating those stem cells, the immunotoxin can allow the engraftment of corrected cells. And in order to do that, we use an experimental setting in which ATM deficient animals were administered with the immunotoxin and then transplanted with cells coming from a genetically identical animal that we can follow up prospectively in order to see the dynamics of the engraftment and evolution. As you can see down there, these animals, when treated with the radiation, same for many other chemotherapy agents, succumb to the treatment, and therefore that is not an option. But then they all survive to the treatment with the immunotoxin, suggesting that it's way less toxic. We have evidence of that, but I'm not gonna show them today. So if we go to the next slide. So by doing so, what we observed is that the immune system is almost fully replaced by 12 to 16 weeks after the transplant. But what is most interesting is in the blue squares under the curve down there is that the speed of recovery of the immune system, both in B and T cells, is much faster in the animals that are deficient for ATM. Moreover, if you go to the next slide, please. Moreover, under the red curve, we basically show in there that some lineages like T cells and like CD4 and CD8 T cells, which are the most deficient in these animals in particular, are able to engraft even without the conditioning regime. What this suggests to us is that the wild-type cells, or the cells that are corrected and have a proficient ATM gene, will have a competitive advantage compared to the ATM deficient cells, and therefore achieving a complete chimerism might be not necessary, but partial chimerisms could be able to actually solve the immune deficiency in these animals. So if we go to my next slide, and I will close here and give the microphone to Mark. I would like to just give you 3 bullet points, and I'll be happy to actually take questions after Mark's part of the presentation as well. We believe that immunotoxin conditioning is safe. Way safer than conventional cytotoxic regimes that allows efficient HSCs, so stem cell depletion and engraftment, and that in models of AT shows that they allow recovery of the immune system on a much faster way with much faster kinetics than in wild-type animals. And with that, I'll close and I'll pass the microphone to Mark.
Excellent. Thank you, Borja. And thank you for inviting us to participate in this event. So we are excited to be here, and we are very excited also to work together with Borja and with Felipe in this project. So actually, we are relatively newcomers to ataxia. So first I'm from Barcelona, Pompeu Fabra University. So our lab is actually very much focused on technology development. So essentially, our main line of research is to create tools that we can do gene transfers with maximum precision and efficiency. And actually, this is what we are using in this project. Essentially, over the last 3-4 years, we have been developing a technology that we are trying to combine the precision of CRISPR. CRISPR, you may be familiar, is this basically molecular scissors that actually was awarded the Nobel Prize in 2020, the chemistry Nobel Prize. But actually, these molecular scissors are very efficient and very precise in finding positions in the genome. But actually, what we are complementing to this CRISPR enzyme is with a gene transfer capacity, so that we can combine the precision of CRISPR with some kind of catalytic center that can allow us to transfer material into the genome. So we can do cut and paste. And here in this slide we can see, right, the find module is this Cas9, this CRISPR enzyme, and then we have this other cut and transfer module, which in our case is a transposase. So basically, it's an enzyme that takes a piece of DNA from a vector and transfers it into the genome. So we have been testing this technology both for in vivo and for ex vivo applications. Next slide, please. So essentially, what are the general properties? So we can get a relatively efficient delivery of genes. We can precisely insert them in specific locations of the genome thanks to the CRISPR. And also another advantage is that it's universal, meaning that since we are inserting the entire gene, so we don't depend on mutation. It's not a type of, let's say, personalized medicine. Next slide, please. And here you have a little animation to show how it works. So basically, we have first the CRISPR enzyme that basically scans the genome until it finds this position where we want to transfer the therapeutic gene. It marks this position, and then basically is where the transposase takes action and transfers the payload. One technical advantage of using this transposase, this component, is that it scales well with size. So basically, we can do transfers of genes that are small and also big without paying too much penalties on efficiency. Actually, the size of the gene that we were working before starting to work into this project is a gene of very similar size as to ITM. We're working with LAMA2, which is involved in a type of muscular dystrophy, and we could get decent gene transfers. Next slide, please. So here is results on this technology on the genome. So basically, when we started combining these two pieces, the Cas9 enzyme, the CRISPR, and the transposase, so you can see all these red dots. So this mean of target. So basically, insertions in places that didn't want. We only want to do the green one. So we had to polish this, and we did this by basically transferring all the capacity of recognizing DNA to the CRISPR enzyme. That is the one that knows how to be precise. And then you can see, for example, in the new version on the right, that basically we only see the insertion in the green point where we are intending to. And this is with, let's say, a precision of about at least 0.1%. Next slide, please. So here I'm just showing some data on cells. So basically, the way how initially we developed this molecular machinery is by basically we can treat the cells and transfer the gene. Here we can compare with a non-programmable technology. This is like the classic transposase that, but specifically is called the piggyback transposase. And actually we get efficiencies comparing our method, which is precise, with let's say the general transposase that are in a similar order of magnitude. So we preserve more or less the efficiency even for large payloads, despite only inserting in that specific position on the genome. And why it's important to only go to this position in the genome? Because we can add then some safety. So basically, we will only insert the gene either in their endogenous loci or in a safe harbor in the genome. Next slide, please. So, and here I'm just showing a little bit what I was mentioning before. So this technology scales relatively well with size. So here, basically in dark purple, I'm showing this, we call it FICAT technology, and compare it to molar recombination, which traditionally has been the classical method for doing precise insertion or precise writing of genes in the genome. And we can see that basically the difference in efficiency, basically the efficiency gap grows with the payload size. Next slide, thank you. Here I'm just also showing just some data, in this case of in vivo. In this case, we're using a lipid nanoparticle, which is adequate a polyplex nanoparticle. We have also used lipid nanoparticles, but this data is on polyplexes. So basically that we can package the molecular machinery plus the DNA that we want to insert in this nanoparticle, transfer it into the bloodstream, and then this basically gets inserted into the liver of the mice, as we see here with this luciferase imaging. Next slide, please. So, and what is the goal of this project? So basically, the goal of this project is to basically apply this technology for delivering the ATM gene in the ex vivo. So basically what we first had to do is to develop a vector that basically will contain the ATM. At the beginning, we'll use a marker, in this case a fluorescence marker, so that we can track the efficiency of this engineering and also how basically stable it is. Right now, we are doing tests in models of bone marrow, like basically cells that we can use easily in the lab, like Jurkat cells, which is a model of T cells, or K562, which is a lymphoblastoid type of cell line. And then as soon as we have results that are ready, we'll move into basically together with Borja to do it in bone marrow of mouse, first of healthy animals, and then disease model animals. Next slide, please. And this is, I think this is the end. Basically from our side, UPF, the people involved in this project, myself and the scientists that are working on this project.
Mark, Borja, thank you both so much. That was really comprehensive, and there are questions coming in, unsurprisingly, that from people on Slido. Do you want to pick up on those?
Yes, we've got a question from Naz Hussain, and Naz, thank you for posing this question. And the question is, how far are we from actually testing this on humans? And I suppose the next part of that would be, and then to actually using this as a treatment.
So, Mark, if you want, I can start and ask from the first side of the
better position.
Basically, the idea of using immunotoxin as conditioning regimes is already being tested in clinical trials. There is one clinical trial that started recruiting a few months ago. It's not going to be on AT or any other primary immunodeficiency in kids, simply because we need to first prove that it's safe enough to use in this kind of diseases. And we hope to have some information about the safety in the next year. From there, it's a matter of doing phase 2, etc., and getting to the clinic if everything goes right.
Yeah. From our side, we have lots of steps before that. So basically, this FICAP technology, first, of course, we need to demonstrate that it works well for ATM gene transfer. But yeah, this technology has been licensed to a company called Integra, and this company will try to use the next 2 years to bring this technology at the level of maturity that then we could start thinking on regulatory clinical phases. But we still need to have all this preclinical validation. First, that it works in ITM, and also to solidify the technology so that then we would be ready to start thinking on a regulatory phase, maybe in 2 years.
Well, Mark
preclinical.
Mark, sorry for interrupting. Mark Borger, I mean, it's encouraging, and that's what we want to show. We want to show the research that is happening. And thank you both so much for the work you're doing, for explaining it so comprehensively as well. And thank you at home, from wherever you're watching as well, for sending in the questions. Keep them coming in, and we'll get as many as we can to all our experts. So that was Mark and Borja. Now, for our second presentation, we've got Dr. Manuela Pellegrini from the Institute of Biochemistry and Cell Biology in Italy. Manuela, it's good to have you with us, and welcome here. Tell us what have you been working on?
Hello, nice to meet you. Hi, Naga. Hi, Mark. So I can start. So of course, it's a real pleasure to be here and to present our most recent work on hematopoietic progenitor cell transplantation and gene therapy for ataxia-telangiectasia disease. So first of all, I would like to thank the AT patients and their family for their support, and also the Italian Foundation for Cancer Research, and also our National Association for Ataxia-telangiectasia. Next, please. So we start our work with the following question. Can we prevent T disease restoring ATM function? Next, please. To answer to this question, we took advantage of a mouse model that recapitulates the measure aspect of the AT disease, except neurodegeneration and telangiectasia. So ATM deficient mice show immunodeficiency, cancer predisposition, mainly B and T cell lymphoma, and sterility, radiosensitivity, and growth retardation. And in our animal facility, the mice die between 4 and 5 months of age. And this is, something to take in account for the next slide. Next, please. So we started our project generating some transgenic mice in which ATM was not functional in all the cells of the body of the mice until we administered tamoxifen hormone following a standardized protocol. After administration, we sacrificed the mice 1 month later, and we checked the phenotypes. What we observed was that ATM treated mice restored the T cell development, looking at the T helper T cells, CD4 single positive cells, when compared to knockout mice or untreated mice, as well as the expression of TCR receptor was rescued. Looking at the spermatogenesis in the lower panel, we can see that after treatment, mice recovered spermatogenesis, and some male mice became again fertile, despite for a short time, when compared to knockout or untreated mice. We then also isolated the thymocytes, and we checked the capability of these cells to respond to DNA damage in vitro, looking at some selected specific ATM target, like CAP1, for example. And what we saw was that the cells were able to respond nicely to DNA damage, phosphorylating the target of ATM, specific target of ATM. Next, please. Next, please. So, a longer observation revealed that the treated mice doubled their survival compared to ATM knockout or not treated mice, and half of them were protected from thymoma, whereas other half undergo thymoma, even though in a delayed way. Next, please. So for the first part, we could conclude that restoration of ATM in young deficient mice is able to prevent or delay all AT phenotypes. Next, please. So bone marrow transplantation can be a good option for AT, as we heard sooner, and basically the possibility to reinsert ATM wild-type cells from an healthy donor into an AT patient could be very important for the disease. Despite this, so far, very few case reports have been reported that actually we recently summarized, outlined in a recent review in Cancers. Despite some success, indeed, there are major lacks. For example, there are not yet patient eligibility protocols, neither transplantation guidelines, and the follow-ups are ongoing. The main concern to use bone marrow transplantation are, first of all, the finding of healthy matched donor for the patients, and also the conditioning side effects, because we need, as we heard, some drug to avoid tissue rejections. And finally, the outcomes are not yet very well known, because these studies are quite recent and very few. So in the lab, we strongly believe that gene therapy could be a valuable alternative to bone marrow transplantation, in which wild-type ATM sequence is inserted in the own cells of the patients, followed by autologous transplantation. So next, please. We started to investigate this strategy. So hematopoietic progenitor cell transplantation was tried in ATM knockout mice. First of all, we collected bone marrow cells from a donor male mouse, and then we enriched these cells with hematopoietic progenitor cells, and that were transplanted into female ATM knockout that were preconditioned to receive the transplantation. After 4, 7, or several months, we look at the phenotype of these mice. And next, please. Blood sampling revealed actually that after 4 weeks, there was an increase in the percentage of the total T cells, and in particular helper T cells, so the CD4 single positive cells. After 7 weeks, the enrichment was more evident, and the percentage actually reached the wild-type cells. Next, please. So by genomic analysis in the upper left panel, we could see that actually ATM wild-type cells reach the lymphoid organs, like in spleen, bone marrow, and thymus, but not in other organs as expected, like tail. And this data was confirmed by the presence of male cells recognized by SRY marker into the female organs, because we did this transplantation, male cells into female mice. So in spleen, we observed that B cell undergo maturation in the middle panels you can see, and basically the percentage of the IgG1 positive cells were restored in these transplanted mice. As well as in the lower panel, we can see that the T cells development was restored after transplantation, looking at the single CD4 positive cells, as well as the TCR beta expression was restored in these cells. In vitro assay demonstrate that after transplantation, we could see a nice expression of ATM in the left upper panel, and also these cells were able to respond to DNA damage in vitro, phosphorylating specific target of ATM. Next, please. A longer observation revealed that the transplanted mice show some sign of aging, like the white fur, compared to wild-type mice, and part of them maintain the T-cell development both in blood and in thymus, and the thymoma was prevented. However, some other mice did not, and they show at some point evident thymoma. According to these results, also the in vitro DNA damage response was observed in some mice, but not in others. So next please. Concluding this part, we can say that hematopoietic progenitor cell transplantation in young ATM knockout mice is able to prevent or at least delay the AT hematopoietic phenotypes. Next, please. What we are working now is a gene therapy strategy. In particular, what we want to do is to insert an ATM wild-type version into lentiviral particles that will be used to infect some hematopoietic progenitor cells that are collected from ATM knockout mice. These cells will be enriched or sorted, and then, as we did before, they will be transplanted into the caudal vein of ATM knockout mice preconditioned, as we did before. After 4 or 7 weeks or several months, we will look at the phenotype of these mice. Next, please. So far we were able to generate a lentivirus plasmid. Yossi Shilo provide this ATM cDNA, and we thank for this. And also we have to thank the EMBL facility in our campus that help in the generation of this lentivirus, carrying a wild ATM version and also a tagged sequence for ATM, the small ultra-red fluorescent protein, and the tags like FRAG and histidine that will help to follow the presence of ATM in vitro and in vivo. We tested the capability of this lentivirus to be expressed both at the RNA levels in the upper panel and also at the protein level in the lower panel. Next, please. Then we were able to produce the lentivirus, so infecting egg cell line, and then we checked the presence of the whole sequence of ATM in the virus through PCR analysis, as we can see in the right upper panel. So ATM was actually present in the virus that we produced. Next, please. We then test the capability of this lentivirus to be integrated and expressed in egg cell line. And actually, after 1 week, we were able to see a nice expression of, so this is actually an encouraging results in this cell line. Next, please. What are our future plans? So first of all, we are now to check the activity of ATM in the cells that have been infected. Then we have to standardize the infection protocols in hematopoietic progenitor cells collected, harvested from ATM knockout mice. And then the final goal will be to transplant these reconstituted hematopoietic progenitor cells into ATM knockout mice to see if we can actually see the rescue of the phenotype. Next, please. So I would like to thank you all, and also I would like to thank all the people that are involved in this project. First of all, Bruna Sabino, our PhD student that did the transplantation part of this project, and also Alessandro Giovinazzo that did a huge work in the preparation and testing of the virus, and Sabrina Putti that performed all the flow cytometry analysis. I would like also to thank our collaborator in Sapienza University and Tor Vergata University, and our, of course, collaborator at EMBL that helped a lot in the making the lentivirus. Thanks.
Manuela, thank you so much. Mark, you've got a question.
Yes, thank you. Manuela, we can see from your work that it's all really around finding a way to get that ATM gene into the bone marrow cell before transplanting that back into the patient. And we've seen some other work like that presented already this afternoon from Marc Güell in Spain, where they used the transposon approach. What's the benefit or the pros and cons of using a virus versus non-viral approaches to get that gene into the cell?
Okay, so it was not very easy actually to make this construct. It takes more than 1 year. And again, I would like to thank the MBL because they did a huge work together with us to make it. We choose lentivirus because lentivirus are already in clinic. So there are a lot of trials ongoing for other primary immunodeficiency, and and also we reach I would say a safety, high safety by using lentivirus. Also today there is a third generation of lentivirus. We use a second generation because it's more efficient, but today there is this third generation that is actually used in clinic. So we think that despite the fact it was not easy to get this plasmid, this lentivirus, but actually is more promising to use in clinic because we know more. But of course, the transposons are also a very good option.
Manuela, thank you so much. Thank you very much for your time and the work you're doing. And again, to all our presenters, there are questions coming in. Time is of the essence here. So what we're going to do, we promise you, any questions that come in, we will pass on to our contributors and get their answers to you. I'm going to move on now to our third presentation from Professor Ernst Wolvetang, who's the Senior Group Leader at AIBN at the University of Queensland in Australia. Now, Ernst and his team have been looking at targeting cellular aging processes in AT. Of course, there is a 10-hour time difference, so Ernst has very kindly pre-recorded his presentation.
Good afternoon, ladies and gentlemen. Cerebellar neurodegeneration is probably the most debilitating aspect of ataxia telangiectasia, but also the least understood one. And today, I would like to show you how we might be able to use human stem cell-derived brain organoid models to not only understand what drives this neurodegeneration, but also that you can use these models to start to screen for novel therapeutics. Before I do that, I would like to thank the organizers for putting on this webinar and for giving us the opportunity to present our work. So how do we make these personalized brain models from AT patients? Well, we start with either a blood sample or a small skin sample, and then we program these cells into what's called induced pluripotent stem cells. Now these stem cells have two important attributes. One, they will grow forever in the dish, so we have an inexhaustible supply of material. And secondly, they can be turned into every single cell type of the human body. And in this case, of course, we are turning them both into neuronal cells, grown in 2 dimensions, as well as in 3-dimensional representations of different parts of the brain called brain organoids. And these brain organoids not only have the cellular diversity and functionality of human brain cells, but also show neuronal activity. And we use these organoids to screen for drugs that might be effective for ataxia telangiectasia. Now, we've been playing this game for quite a while now, and we were the first to make induced pluripotent stem cells from ataxia telangiectasia patients, the first to gene correct the ATM gene in these stem cells, and to study the gene regulatory networks in cerebellar neurons that are degenerating in AT. What I would like to do today is to tell you about the latest story where we use these models to start to understand neurodegeneration in AT. Now, it's been known for some time that cells from AT patients or from AT mouse models show many of the features that are also observed during normal biological aging. And of course, this starts with genome instability, which is not surprising for a disease that lacks a protein critically involved in DNA repair, but it has now also become clear that many other aspects of the aging process, including cellular senescence and mitochondrial dysfunction, are key features of ataxia telangiectasia. And it is these aspects that I'm going to talk to you about today. So let's start with mitochondria. As you may know, mitochondria are the powerhouses of the cell, and you could compare them to the motor that drives everything that's going on in the cell. And like a motor, it has a clutch as well as an exhaust. In this case, mitochondrial membrane potential is a measure of how well the clutch is engaged. And when Dr. Hannah Leeson, a postdoc in my laboratory, measured mitochondrial membrane potential in neurons derived from the pluripotent stem cells or in primary cells derived from the nose from patients with AT, she could observe a reduction in mitochondrial membrane potential. And at the same time, these cells produced more reactive oxygen species. So in other words, the exhaust fumes of the motor, which are bad for both the cells themselves as well as the DNA that they carry. At the same time, one of our close collaborators, Professor Martin Levin, who has been working on AT for the last 25 years, showed that in cells where ATM is deficient, mitochondria are also dysfunctional. And importantly, he was able to show that when he added triheptanoin, which is a fatty acid with 7 carbon atoms, that the survival of these ATM deficient cells was enhanced. And excitingly, this has now led to the first clinical trial here in Australia for children with AT, which is led by Professor David Coleman, who is here triumphantly holding up a bottle of triheptanoin, which took a long time to get into the country during the COVID crisis. But mitochondria are certainly not the entire story if we talk about neurodegeneration in AT. And an important clue was found when a postdoctoral researcher in my lab, Dr. Julio Aguado, started to make brain organoids from these pluripotent stem cells from children with AT. And what he noticed is that these brain organoids were much smaller than their wild-type counterparts. And not only that, when he measured the number of senescent cells in these brain organoids, he noticed a pronounced increase in senescent cells in the brain organoids. This was surprising to us because senescence is normally associated with the aging process, and in this case with brain cells that are coming from AT children. So this led Julio to investigate what was actually driving this increase in senescence in AT brain organoids. And to make a long story short, what he was able to show is that a particular signaling pathway called the cGAS-STING pathway was the pathway that was responsible for the increased senescence in the AT brain organoids. Now the cGAS-STING pathway is a sort of cellular immune system that detects foreign DNA or DNA that is at the wrong spot in a cell, and depending on that will activate genes that increase the production of proteins that induce cellular senescence. Now, what Julio was also able to show is that inhibitors of this cGAS-STING pathway, in this case either aspirin or a compound called H151, was able to rescue both the size reduction in the AT brain organoids as well as the senescence that we observed, suggesting again that the cGAS-STING pathway is the one responsible for increased senescence. He then started to dig in further and was able to show that this cGAS-STING pathway, or that inhibition of this cGAS-STING pathway, was able to restore the amount of neurons in the brain organoids from AT children and reduce the abnormal amount of astrocytes that we observed in the brain organoids. And he also showed that when he added these cGAS-STING inhibitors, that the activity of the neurons was also restored, suggesting that cGAS-STING inhibitors may have therapeutic benefit for inhibiting neurodegeneration in AT. So how does this all work? Well, it's complicated, but let me see if I can explain it to you. So what we think is happening is that in AT, because of the absence of a protein that repairs DNA, DNA fragments leak out of the nucleus of a cell, and together with DNA released from dysfunctional mitochondria, activates the cGAS-STING pathway to activate transcription factors that upregulate what's called SASP factors, or senescence-associated secretory proteins. These are the proteins that are released by cells to induce senescence in neighboring cells. And what we were able to show is that these SASP factors are particularly produced by astrocytes, and that these dysfunctional astrocytes lead to dysfunctional neurons and neurodegeneration. So this means that cGAS-STING inhibitors may be able to inhibit SASP in AT patients and then improve neuronal survival and activity. Now, another way to get rid of senescent cells is not just to prevent their emergence, but also to get rid of them after the fact. And one way to do that is to use compounds that are called senolytics. And what Julia was able to show is that if we look at the amount of senescent cells in the brain organoid from AT children, that exposure of these brain organoids to different senolytics was able to reduce the amount of senescence cells in these brain organoids, and at the same time increase the expression of lamin B1, which is another indicator of senescence. So this suggests that not only inhibiting the emergence of senescent cells, but also getting rid of already senescent cells could be of benefit to AT patients. So what does this all mean in the wider sense? Well, one of the things that we are starting to realize is that there are many similarities and parallels between normal biological aging and ataxia telangiectasia, which extend well beyond what we see in the brain. So in the brain, we now think that senescence is a strong contributor to the phenotype and neurodegeneration and ataxia telangiectasia, but it is also possible that increased cellular aging affects the eyes, the heart, the fatty liver phenotype, the gonadal dysfunction, and other phenotypes that are seen both in AT and in aging, such as immunodeficiency and diabetes. And in a way, we are starting to think that AT might be another example of an accelerated aging disease, and maybe should be treated accordingly. The take-home message of what I've told you today is that not only can we model ataxia telangiectasia brain degeneration in brain organoids derived from human stem cells, but significantly that in AT there is strong features of accelerated aging in brain cells observed in a dish, and that we can use inhibitors of the cGAS-STING pathway as well as senolytics to get rid of these senescent cells in AT brain organoids, suggesting that they may be of therapeutic benefit. And I also told you of our ongoing clinical trial to improve mitochondrial function with the C7 fatty acid triheptanoin, and this trial is supported by our patient advocacy organization, Brushshot. So are we ready? Well, It may be too early for clinical trials for AT with senolytics or STING drugs at the moment, but there are ongoing clinical trials with senolytics for Alzheimer's disease and kidney disease, and it appears these senolytics are well tolerated and relatively safe. However, in the case of CGAS STING inhibitors, we cannot rely on using aspirin because the dose is just too much to be clinically relevant. So we are waiting for a number of drug developers who are at the moment making more specific and better tolerated CGAS STING inhibitors. And perhaps at that moment in time, we could start to consider combination therapies of senolytics and CGAS STING inhibitors and see if they work in terms of inhibiting the neurodegeneration that is plaguing our children with AT. And with that, I would like to conclude my talk, thank my fantastic team here in Brisbane, the robot for working 24/7 in screening drugs for AT, our funding bodies, but most importantly Brushut, for their enthusiastic and continuing support for our work. Thank you for your attention.
It's always good to thank the robot. Of course, because of the time difference, Ernst was unable to join us right now, but he has been answering questions via Slido. He's unable to answer your questions via Slido, but he has been able to answer some questions from Mark, who was able to catch up with him a little earlier, and asked to explain senescence in AT.
Senescence is something that we all know very well because we see ourselves turning grey and we see our skins wrinkle as we age. But what we don't see is the senescence that happens in our brain cells, which happens both in normal aging, but also now, as we've shown, in an accelerated way in children with AT, or at least in our model. So the problem with senescence is that they're called zombie cells, not for nothing, because they can actually tell their neighboring cells to become senescent as well.
So if treatments can be developed to slow down this acceleration in the aging process, what might that mean for patients with AT? What would they experience? How could their symptoms improve?
In terms of its relevance to AT, therefore, particularly slowing down this accelerated aging or senescence process in the brain of children with AT is hopefully going to slow down the neurodegeneration, but it's unlikely to reverse the damage that has already been done. And it is this slowing down of senescence in the brain that we're hoping to achieve using these targeted SENO-STING therapeutics.
So could this actually reverse damage?
I expect that senolytics and senostatics and CGAS STING inhibitors are likely to affect the premature aging of the brain in AT. I don't think they will reverse the pathology, but they are hopefully going to slow down the neurodegenerative phenotype that we see in children with AT.
So your lab are currently waiting for the development of specific CGAS STING drugs before being able to embark on the possibility of clinical trials. Do the developers have an idea of a timeline and when they realistically think these improvements might be made?
In terms of the CGAS STING inhibitors, they are still a couple of years off to finalize their developments, and we're keeping close eye and are in communication with the various companies that are doing that. Once they are ready and tested in animal models, then we certainly will need to do clinical trials in cohorts of children with AT. And depending on the jurisdiction, this could be done on compassionate ground in small numbers, but ideally would be a multi-center international trial with multiple participants across different countries. And organizations such as Action for AT and Brushut would certainly play an important role in coordinating these efforts. And that would be a fantastic thing to see happening.
So the senolytic clinical trials so far are in Alzheimer's and kidney disease. If these trials show that the drugs work in those diseases, how long might it be before we can trial those drugs in AT, or even perhaps get approval for use in that condition?
At this stage, we don't know if senescence also contributes to disease that occurs in other organs in AT outside of the brain. But at the moment, we are investigating this in liver organoids, and it would be exciting if senolytics or cGAS-STING inhibitors could also improve the problems that we have seen in AT there. But we don't know at this stage.
As the current studies are in adults, what extra work may be done before those trials could be done to test treatment in children? Are any other paediatric diseases trialling or using Seno-STING drugs?
At this stage, we are not aware of other clinical trials that are using Seno-STING inhibitor pathways, certainly not for children. I think we should be particularly careful in making sure that these drugs for AT children are safe. And I would like to see animal studies and safety studies done first in children with AT, because as we know, they have a whole range of other problems that may be exacerbated by these inhibitors. So we have to be very, very careful before we move ahead with major clinical trials.
Mark, thanks very much for those questions. And Ernst, of course, our thanks go to him too. This does conclude the first half of our research presentations. I hope that you're finding the content interesting and looking forward to hearing from our second half speakers. We are going to take a short break now, just about 5 minutes. We'll be back for the final presentations then. And during the break, we'll run our second poll to help understand which areas of research you would like to see funded more. So do take part on Slido. Use the poll button in the top right-hand corner of your screen. We'd like you to select one of the following themes: metabolism, gene therapy, drug repurposing, biomarkers, or others. Now, you saw 100% on the top, but that's only just because only one person had voted by then. So I need lots of votes coming in so we can get a really good, true picture of what you think. We shall see you in around 5 minutes. Now, welcome back to the second half of the Spotlight on AT Research webinar. We still have 3 really good presentations lined up for you. Before we introduce the next speaker, just want to check out the results of the poll. Thank you very much for taking part. Mark, we've had an interesting look at this, haven't we? Has anything surprised you from the results?
Yeah, actually, it's not a huge surprise. It's great to get this input from the audience so we can target where we put our funds and the charities can think carefully about where they go. But it's no surprise that with a genetically mediated disease like this, that gene therapy is the top choice there. This really is the thing that if we can correct will make the most difference. Drug repurposing also came second, and again, no surprise, because that's all about using drugs that are available, but testing them and then using them more quickly than perhaps developing new drugs to treat these patients. So
time is of the essence, isn't it, obviously. Mark, thanks very much. And thank you very much as well for providing your views. As Mark said, the Global Alliance members will consider your feedback when making future funding decisions. Now, we're going to return to our research presentations. First, we have Lisa Bunn and Munira Khan from the University of Plymouth, who are going to talk about producing healthcare guidance for children and young people with AT. Munira, Lisa, hello. Thank you very much for joining us. Please begin.
Thank you, Naga. So hello, and thank you for inviting us today to talk about this really important project, producing healthcare guidance for children and young people with AT. My name is Dr. Lisa Bunn, and Munira Khan and I are here today to represent a relatively large team of people who all work together on this project, and we're collectively known as the A-Team Collaborative. The project's been funded by Action for AT, as well as being significantly supported to date with recruitment of participants and involvement and engagement activities by the AT Society here in the UK. So the studies we'll talk about within this presentation have been coordinated and predominantly undertaken by Munira Khan as part of her PhD study. And before we start, we really want to acknowledge the wider team's contribution to the slides that we will present today. Next slide, please. So the A-Team is a group representing children and young people with AT. It includes family members, therapists, nurses, clinicians and researchers, all working together to understand how therapies can improve health and everyday life. We use the term therapy to describe nursing and allied health professional contributions, rather than more widely research fields of medical, pharmaceutical and surgical therapies. We're particularly interested in therapies that could be done at home or at school. Therapies that could help keep people with AT in good general health and optimise fitness, so that when extra AT-related complications occur, such as respiratory tract infections, a reduction in mobility level, or surgery for tumour resection, we can facilitate optimal recovery, rehabilitation, or help people adapt to keep engaged with the things that matter most to them in everyday life. We're ultimately trying to achieve a good understanding of how allied healthcare can contribute to the management of AT. We want to offer better evidence-based guidance and contribute to that growing evidence base with our own randomised controlled trial, an RCT of a pragmatic home-based intervention. Next slide, please. So in order to achieve our aim to improve health in everyday life with allied health therapies, this project sets out to achieve 3 main objectives. First, to produce up-to-date allied healthcare guidance, and in a moment, Munira will present our findings of a recent extensive scoping review of the evidence aligned to this objective. The second is to work with children and young people with AT and their families to produce patient-held health records that are owned by children and young people and their families and can be shared with others who might benefit from reading these personalised notes, such as GPs, physiotherapists in the community, maybe PE teachers, dietitians, or community nursing teams. Our initial engagement activities with families suggested that there's considerable time wasted having to recap key information about AT, which could otherwise be better spent dealing with the reason that they'd made the consultation in the first place. So by helping people with AT to produce their own sets of notes to share and own, hopefully this will be empowering and save time in already very busy everyday lives. Our third objective is to work with children and young and families to develop new exercise-based interventions, which can be done at home or at school or in a local gymnasium, which can potentially improve fitness and everyday health and help to maintain this over time. We intend for this home-based intervention to enable families to connect with other families with AT too, using digital technology. So regardless of geographical location, families will be able to connect and offer support to each other within the exercise programs. From our personal experiences and professional experiences within the group, as well as ongoing PPIE, we're very aware that exercise can be an opportunity to improve fitness and have fun, but this also takes time and energy. It can be difficult and tiring, and as such, it can also be a burden. Before we launch into a fully powered randomised controlled trial to test whether or not the intervention that we design is effective or not, we'll first conduct a slightly smaller trial to evaluate how enjoyable and feasible it is and make sure that it really is safe to do at home. Next slide, please. So, this project started by and continues to work with children and families with AT. Their experience of therapies and allied healthcare to date, their ideas about how things could be improved in the future, and their preference about how this project proceeds and evolves. This close working and development of relationships over the project duration is known as co-production of research. It's slightly different to a concept known as patient and public involvement and engagement because it really commits and strives to offer joint ownership of research and a shared understanding. Offers shares control over the research process, mutual value and respect, and it really includes and not just involves patients and families. Really importantly, it also gives opportunities to learn and reflect on shared experiences, unlocking everybody's individual potential in the group. Next slide, please. And over to Munira.
Thank you, Lisa. So the objectives of this project will be achieved by undertaking 4 work packages. The first work package included synthesizing evidence on care and management of children and young people with AT provided by nurses and allied health professionals. Considering the type of evidence that we were exploring, its variability was quite broad and diverse, and therefore a scoping review was undertaken. Our focus was on children and young people with AT who were less than or equal to 18 years of age. We also looked at other inherited and progressive ataxia types such as Friedreich's ataxia, ataxic cerebral palsy, and others in order to consider all available key transferable knowledge. We excluded any medical and pharmacological interventions and psychological care and only focused on allied health and nursing therapies. We used the Joanna Briggs Institute approach for conducting the review and used many different databases to search evidence, including those that list journal articles, clinical trials, conference proceedings, and also looked at the Gray literature sources. Over 16,000 sources of evidence were initially identified, but after removing the duplicates that were identified from different sources, over 14,000 sources of evidence were selected for screening. A vast majority of them were not relevant to our review, as they were either focused on adults, they didn't look any interventions or therapeutic interventions. They were perhaps looking at an experimental type of treatment that didn't involve any additional exercise or conventional type of therapy. After consultation with the experts in our team, 38 sources of evidence were selected for final review. 22 of those looked at interventions and documented the impact of those before and after using the outcome measures. 1 of them looked at the experience of undertaking a trial. 3 of them used both approaches and reported a bit of each, and 12 of them were non-research materials like guidelines, reviews, and practice documents. Next slide, please. The results of this review revealed that some of the interventions were reported with significant effects, and particularly interesting within AT-specific literature was therapeutic exercises, including coordinative training, inspiratory muscle training, and nutritional intervention, including early use of PEG devices to supplement ingestion of food. However, no formal recommendation for practice can be made yet because of the lack of quality of research undertaken to date, and there is a very clear and urgent need for more robust research. The formal write-up of this piece of work is currently in submission with one of the peer-reviewed academic journals. If you would like to look at a more comprehensive protocol that was written guiding how we undertook this particular review, this is available open access at the A-Team Collaborative Open Science Framework website on the link given at the bottom of the slide. Next slide, please. The next work package includes co-production of 80 specific patient-held healthcare records, which we are provisionally calling My18 Pack. This will be owned and held by children and young people with AT and their families. We hope it will provide capacity to track symptoms, record activities related to therapy interventions, and help with information sharing with different professional groups. However, the final content of the pack will be informed by the needs and requirements of children and their families. To co-produce this pack, we have prepared a dedicated online web page. It's private and secure, available for participants to sign up on personal computers or smart devices. It will provide a platform to join forums using secure and private chat room options and allow people to share files that may be of relevance to co-production of the My18 pack. Focus group discussions will be conducted with children and young people and their parents or carers. And hopefully by the end of the study, we will have a digital template of the pack for the participants to download and use. And we're also hoping that some of these participants might help us to also co-produce an intervention study, but it's not a requisite for participation in this study. Next slide, please, and over to Lisa.
Thanks. So in order to develop a successful intervention study, Munira is undertaking workshops with children and families with AT to direct final design decisions. But we've also had a very clear steer from the evidence base. And we think that the intervention needs to have some home-based respiratory and whole body exercise components. We intend to have sessions pre-recorded that are aimed at being engaging, yoga-based exercises. And these have been provided from Cosmic Kids Yoga. So using our new and secure webpage, we can select different movies for each individual participant from an existing bank, so that we can really target people's training according to age and ability. And the dedicated webpage and the team involved will also allow us to offer support when needed from both neurological and respiratory physiotherapists, as well as being able to allow us to help troubleshoot problems and, of course, importantly, monitor any adverse events. Next slide, please. So we're hoping to integrate our AT pack into the assessment in order to one day evaluate effectiveness of the programme. But there are also some standard validated outcome measures that we can adopt within the study, which will allow us to look at the impact on ataxia, disability, respiratory muscle function, and overall quality of life. It's also really important to assess the ability to enjoy and be confident about undertaking exercise. So we'll be producing bespoke daily diaries to make sure that we have an understanding of how acceptable and burdensome the intervention might be on a daily basis. We want to find out about feasibility and how easy it is to adhere to. The way that we produce and stream the exercise movies through the webpage will allow us to collect measures of how often and how long for the movies have been watched for. So it will give us another way, in addition to the diary, in order to find out how easy it is to engage with the intervention. Next slide, please. So in summary, this project set out to collate best evidence-based practice guidelines for management of AT. It aims to offer the first AT-specific patient health notes owned by children and their families. It will evaluate the feasibility of a home-based exercise intervention and conduct an onward RCT if feasibility and safety are first established. Next slide, please, and over to Munira.
Thank you. So how can families get involved? Well, anyone can join the AT. It's a funded project, but members are voluntarily participating with no payment. Names can either be listed on our open science framework or not, according to your preference. And the only key requirement for participation is an interest in therapeutic management of AT. Families can also actively participate in the program of research. We are currently recruiting to the My18 work package, so if anyone is interested in participating, please contact me or Lisa on the given email addresses for more details. Next slide, please. And I would like to take this opportunity to thank our funders, Action for AT, and to all of the AT members who have contributed their time to the project to date, as well as the organizations who have supported AT members' time. We welcome any questions either now or later by correspondence. Thank you very much for listening.
Munira, Lisa, thank you both so much. Let me show you and read to you a lovely comment that we've had on Slido for you. It says, it's from Anonymous, so they haven't identified themselves, but just saying, can I congratulate this team? Because it seems that this may help families soon, and there are not many things in the pipeline that can do that. Thank you. And you know, of course, you can understand the frustration of so many families who are listening today, who are seeing the developments in science, but these things take time. So the fact that you're presenting something practical is offering some comfort as well for many of those watching. So thank you. Mark, did you have a question?
Yes, a quick question, if that's okay. So you described very well how when you did went to do your review, the scoping review, there wasn't enough robust evidence there to actually allow us to pull together some robust guidelines for treatment in this type of therapeutic area. So what do we need to do? What needs to be done to get to the point where we can have some robust guidelines for this type of non-pharmacological therapy? Is this a call to action?
It absolutely is a call to action. More research is needed. More research is needed across the board in AT, I feel, but the therapeutic research should not be neglected. Physiotherapists, OTs, dietitians, to name just a few, have a really important role in the management of AT. And provide a really significant contribution in the absence of a cure presently. But we are working on a case-by-case basis. We're trying our hardest to use good clinical reasoning, but what we really need is to know whether treatment approaches work or not. And the only way that we'll get that knowledge is to have well-designed clinical research.
And Munira, thank you as well very much. And for anyone who is watching, you did see Munira and Lisa put their email addresses up on screen. But of course, if you have any questions, you put them through on Slido. We will make sure they are passed on, as we will do to all our contributors. We're going to move on to our penultimate presentation from Svetlana Korenkova of the University of Cambridge, who's asking the question, why do AT neurons die? Svetlana has pre-recorded her talk, and we're hoping that she'll be able to join us live, technical gremlins willing, straight after this presentation.
Good afternoon, everyone. I'm really excited to be here, so thank you so much for inviting me. And I'm very sorry I'm not presenting live, but I will be with you in just a few minutes for the Q&A session. So my group at the University of Cambridge works on investigating mechanisms of neurodegeneration in genetic diseases, and we're specifically interested in ataxia telangiectasia. Now, neurodegeneration in AT, which is a really devastating aspect of this disease, stems from the loss of a very specific type of neurons called Purkinje neurons in the cerebellum. So here you can see an image, MRI scan actually, image of the cerebellum from a healthy individual, and also an individual with AT. And the loss of matter in the cerebellum in AT is very apparent. It's almost like a cerebellum loses weight. And it is responsible for this devastating loss of coordination phenotypes that brave little kids with AT start experiencing often in their toddler years. And this is just an image of a Purkinje neuron, very pretty neuron, that actually dies in this disease. The fascinating fact about neuronal loss in AT is that AT neurons do not die by themselves. So if one takes a neuron from a healthy individual, so healthy neurons, and put them on a dish, which is something we can do in the lab, then these cells sustain themselves happily. We can now take neurons from an individual with AT or model AT neurons in the lab, and these neurons would sustain themselves pretty much as happy as healthy neurons. Now, if we take a mouse in which ATM is deleted in neurons specifically, then we actually do not observe neuronal cell death. And this is very recent work from Peter McKinnon lab. And if we now take a fly and genetically modify it so that ATM is deleted in all cells in the brain, then this fly would have neurodegenerative phenotype, so they actually have loss of coordination. Now, if we take a fly and remove ATM in neurons only, then these flies actually do not have neurodegenerative phenotype. And it was the Weissman lab who nailed it, because what they've done, they deleted ATM in glial cells in the brain, and they observed loss of neurons and also glia and neurodegeneration. So what it effectively means is that glial cells somehow harm neurons in AT. There are lots of different types of glial cells, just like there are many types of neurons in the human brain. But if we look at the levels of ATM protein in different cell types in the human cerebellum, which are shown just here on the left, then we find that ATM actually has the highest levels in cells called microglia. So now if I can talk you through this graph, so the size of the circle simply shows the percentage of cells of a specific cell type in which ATM protein is present. And the redness of the circle, so this is the scale, the redder the circle is, the higher the level of ATM protein in a specific cell is. So this big red circle in this case means that ATM expression, ATM levels are the highest in microglia. And this data come from incredibly timely and resourceful piece of work from the labs of Alice Lee and also Mike Lodato in the US, who basically performed sequencing of cerebellar tissues from healthy individuals and individuals with AT. And another fascinating thing is that the levels of ATM in Purkinje neurons, those that die in AT, are very low. What this means is that ATM must be very important in these microglial cells. And now I can tell you a little bit more about microglia. So microglia are incredibly important for keeping the brain healthy. And as a part of this function, microglia detect and remove, by effectively eating, dying and damaged and also dead neurons and other cells. And if dead and dying cells are not removed, then they release lots of different compounds that can be toxic to healthy cells around them. So microglia are performing pretty important function. And in neurodegenerative diseases, if microglial function is affected, they can actually cause harm by either not removing dying and dead cells very efficiently, and which would result in toxicity, or by being overactivated and removing not just dying cells but also healthy neurons that shouldn't be removed. And here is just a little video of a microglia on a dish, which fills the environment around it and moving around and eating green beads that mimic dying neurons. We think that AT microglia, as I already mentioned, are dysfunctional. They become hyperactivated, and this hyperactivation happens because loss of ATM protein leads to damage to our DNA, to our genetic information. Such damage results in loss of normal regulation of microglial function, and they start killing neurons by eating them excessively, but also by producing various toxic compounds during this activity. Now, the main questions that we aim to ask in our research project include this one, which is: how do AT microglia become hyperactivated? What is the mechanism and what is the sequence of events? Now, as I already mentioned, loss of ATM leads to changes in our genetic material. And this damage to our genetic material drives microglial activation. And we really want to know molecular pathways that drive this activation in detail. We've identified one, but obviously we want to know more details of how it happens. Now, activation of microglia leads to production of toxic compounds that actually get released outside the cell. These compounds damage healthy neurons and other microglia and other cell types around them, but they also activate various proteins on the surface of microglia. Now, this activation of these proteins on the surface of microglia actually leads to further activation of microglia and their dysfunctional behavior. And we basically create a vicious circle of microglia becoming more and more activated and more dysfunctional. And our aim here is to understand the details of these events so that we can modulate the activity of this pathway and hopefully bring benefit to individuals with AT by informing some novel strategies for treatment of the disease. And what we would like to do is we would like to concentrate on trying to block some of these surface proteins and basically prevent further activation and dysfunctional behavior of microglia. Now, to block some surface proteins, we actually need to know what. And what we did is we collaborated with Alice Lee and Jenny Lai and of course Michael Odato, who provided the sequencing data that I already mentioned in some previous slides. And we looked at the amounts of some of the relevant surface proteins in AT versus healthy controls, specifically in cerebellum microglia. And the principle is the same as before. We're looking for large dots, and these dots have to be red or orange to show increase in the levels of specific proteins. And you can see that at least for these 5 proteins, we have fairly low levels in fairly low percentage of microglia in healthy brain, and we have quite a lot more of these proteins in AT microglia. So these proteins are potential targets for our further investigation, and we're validating now that these proteins are indeed activated and play a role in microglial dysfunction in AT versus control microglia in a dish, and we will further attempt to modulate microglial dysfunction by blocking the action of these proteins. Or set of questions that we want to ask is how precisely AT microglia kill neurons and what kind of neurons do they kill? Because, of course, if we know mechanistic details of the process, we can actually think how to reverse it correctly without inducing various side effects, which is incredibly important in the clinic. And here I want to show you some images of neurons on a dish. So this image shows the neuronal network. It's effectively a spider net that neurons use to communicate with each other and send signals from one to another. And it's colored in magenta. And you can also see some blue dots, which shows genetic information of these neurons. And this is how a healthy network of AT neurons looks like. Now, if we take AT microglia and put them on top of AT neurons and let them work effectively with each other, then what we see is actually lots of damage to the neuronal network, which you can see here. It looks like Pac-Man's been here. Good old days when we played these games. But anyway, if you look on the right, so this image shows a merge image of both AT microglia, which are shown individually here on the left, and they're shown here in white, and also this neuronal network, so, you know, the connections between neurons that are used for communication, and they're shown here in magenta and on this image in white. And what you could see is that we have a formation of a so-called cluster of AT microglia. and this cluster co-localizes, so it's in the same place where the damage to neural network is. And we think that this cluster of microglia actually causes the damage by eating excessively everything that they meet. So if we now zoom in to this square and look at this cluster in more details, then what we could see is we see these holes inside microglia that are actually filled with neuronal material. So what happens is that microglia, as I already said, eat neurons, and they end up in these so-called phago lysosomes, which are effectively stomachs of microglia where they digest what they've eaten. And we find that they eat dying neurons, which is a normal function of microglia, but also absolutely healthy neurons, which we think shouldn't be eaten, and also pieces of neuronal network. And what we're doing now is we're continuing to investigate these events and also the origins of microglial dysfunction to improve our knowledge of neurodegeneration in AT, and also to hopefully, as I already said, inform the development of novel treatments for this devastating disease. So I'm going to stop. Thank, first of all, the AT Society, which is very kindly funding this work. I would like to say thank you to group members of my lab who contributed to the work on microglial dysfunction in AT until now, and to our collaborators, Jenny Lai and Alice Lee and Michael Lodato in his lab, and also Tom Chittenden, Genuity Science in the US, who help us with bioinformatic analysis of sequencing data. And finally, this is an image of Lisha Joshi, who is a new postdoctoral scientist in my lab who has just started working on this project. And thank you very much for your attention, and I look forward to your questions.
Svetlana, thank you so much. And I think we can, yes, there she is. Svetlana, good to have you with us on Monitor. It was a really fascinating presentation. There will be lots of interest in your work, as neurodegeneration was highlighted as a key research theme at the previous Spotlight on AT seminar that we held, the webinar that we held. Just a reminder to you before we get some questions to Svetlana, if you can get your questions via Slido, we can see them in front of the screen in front of us, and we can just throw them at her and get as many answers as we can. Mark, I know you've got a couple of things you want to ask Svetlana.
Yes, thank you. And I think the first one is really, it's very similar to one that we have in Slido actually, which is from Margarita Doria. And Margarita says, may non-cell autonomous external factors take part in the microglial activation? And in addition to that, I was going to also ask, is there any other factor you think might be taking part in the death of Purkinje cells? Are there other things that we ought to think about researching alongside your work?
Thank you very much, Mark, and thank you, Margarita, for this wonderful question. So non-autonomous cell factors, so what it means is that the origin of these factors would not be within microglia, but in some other cells around them, of which, we know of many different ones. So Ernest, for example, talked a little bit about astrocytes that are dysfunctional in AT. And we also know that there are also problems with Purkinje cells themselves. The question is whether these problems with Purkinje cells, which are effectively problems with producing proteins required for their normal function, whether those problems actually translate into the death of neurons. So we think that microglia are really important to detect various defects in cells around them. And in addition, they are themselves dysfunctional, and altogether it drives the loss of neurons. So I hope I'm answering both questions at the same time, actually, Mark. If not, please come back to me.
Yeah, no, that's great. Thank you. And how long do you think it will take to get some of the answers to your questions, the research that you're doing?
Well, we've started this project, so it is based on some preliminary work that we've been doing over the last 2-3 years in my laboratory, and hopefully within the next couple of years, we will have more information on microglial dysfunction. And just to clarify, it's not just microglial dysfunction, it's the dysfunction of astrocytes, which is another cell type, glial cell type in the brain, and some other things that need to kind of come together to provide therapeutical benefit to patients with AT. So overall, this process is not going to be immediate. It's not, we're not talking 2-3 years, but in 2-3 years, hopefully we'll have more molecular mechanisms and details that should help us inform thinking about the therapies.
That's very clear, thank you. And perhaps one more question. This is from an anonymous person. They haven't popped their name in, but it's received a vote as well. Would the processes that you're researching be the same for other cerebellar ataxias? For example, ataxia with oculomotor apraxia type 1, for example?
This is absolutely a great question. And this is what we are starting to think about because effectively, I already mentioned in my slides that we think it's not the loss of ATM itself that drives microglial dysfunction, but it's the damage to our genome that loss of ATM causes that actually contributes to this abnormal functioning of microglia. So I would expect that to a certain extent, at least, it would be very similar in a range of different neurodegenerative disorders that are linked with genome instability. And it would be incredibly exciting to look into it and, find some answers. And perhaps we could eventually target not just individuals with AT, but many, many more kids with neurodegenerative diseases.
Of course, that is the hope. Svetlana, thank you so much. Thank you very much for the time you've taken to present today. There are more questions, and as promised, we will get these questions to all our contributors, to all our presenters, and get their answers to you. And of course, all their presentations are going to be made available after today. We're going to move on to the last presentation of the day from Taylor Fields, Chief Product Development Officer and Senior Vice President of Intro Bio Limited. Taylor, good to see you. Thanks very much for joining us. Last but by certainly means not least.
Thanks. So it's a pleasure to be able to present for you all today, and I'm here to give an update on InterBio's development program for ataxia-telangiectasia. Next slide, please. So InterBio is currently developing a drug called N-acetyl-leucine for the treatment of AT. N-acetyl-leucine is a chemically modified amino acid, and it can be formulated as either a racemic mixture or the individual L and D enantiomers. For any non-scientific background people, I always describe it to patients and families as you have your left and right hand and you can cross your hands. Now, they may look similar, but they function differently, just like these drugs function differently in the body. So they really are three distinct molecules. Our initial development had begun with the racemic mixture, which had been approved in France in the 1950s for acute vertigo. It is not approved anywhere else in the world for any other indication, but has over the last 60 years acquired a very benign safety profile. And it was because of this safety profile and what was then believed to be the mechanism of action that our collaborators at the University of Munich had the idea to commence compassionate use studies some 10 years ago. And over the past 10 years in these observational compassionate use studies, we've observed both the symptomatic and disease-modifying neuroprotective effect of this drug for over 20 different neurological disorders, including AT. Next slide, please. So in parallel, our scientific founders at the University of Oxford have been doing a litany of preclinical studies and have identified that the L enantiomer, N-acetyl-L-leucine, is the active ingredient in that racemic mixture and has superior safety and efficacy when administered independently. It's also an orally administered treatment. It has a very benign safety profile. And this L enantiomer, which we call IB1001, is not available anywhere in the world. I'll get to this in a moment, but we know that the drug is transported both across the blood-brain barrier as well as to other major organ systems. And again, we believe it has broad applicability, both in regards to symptomatic but long-term treatment for disorders like AT. Next slide, please. So the very simplistic overview of part of the mechanism, but we sometimes are asked what's the difference between N-acetyl L-leucine and L-leucine, which you could buy as a supplement. Essentially, that acetylation transforms L-leucine into a drug. So N-acetyl L-leucine is a prodrug for L-leucine, and that acetylation is hugely important in regards to its transport mechanism. So traditionally, L-leucine is transported throughout the body by an amino acid transporter. When you add this acetyl group to it, it allows it to be picked up by the monocarboxylic transporter, and essentially delivers it in quantities, much higher quantities, quantities that you could never get with L-leucine. So we've published quite extensively on this. Again, I've had limited time today, and it's a very brief overview, but I wanted to just highlight that this unique transport mechanism is intrinsic to its pharmacological activity, and it's the acetylation of this drug which is allowing it to have its clinical effects. Next slide, please. So once the drug has been delivered, transported and delivered within the cells, we know that it actually has multi-pharmacological effects. So it's somewhat functioning as a combination therapy in its own right. We believe that its primary effect is in the normalization of energy production. So we see a huge increase in ATP production, which in the mitochondria, which has direct knock-on effects in the lysosome. So we see normalization of lysosomal volume and the accumulation of lipids. These might not be as directly relevant for AT as they are in some of the other lysosomal storage disorders that we treat, but that correction of energy metabolism is definitely important. We're also seeing a stabilization of neuronal membrane potential, and we see a huge dampening of neural inflammation. What you see on the bottom here is some results from the preclinical in vivo studies in the Niemann-Pick disease type C model, which actually is a very good model for AT in regards to its clinical presentation. And we're seeing both a significant expansion of lifespan, but also what you see on the bottom right here is when we treat these mice either pre-symptomatically or acutely when they're symptomatic, we see a very significant reduction in ataxia, in gait dysfunction, and motor impairment. And so we're seeing that if even pre-treatment or when mice are already symptomatic, a significant improvement in these clinical symptoms. And that's also what we see in the clinic. So if we can go to the next slide, please. So the video playing now is just the age-matched treated and untreated MPC null mice. The one with a noticeably better coat condition is the treated mouse. It also is much less ataxic, has much more improved mobility, and not pictured, lives quite a bit longer than the untreated mouse. So I think it's just good to see the illustration of what these graphs are showing us. Next slide, please. So for AT, we've completed an observational case series of 6 patients, and we looked at each patient at a baseline period, after a 1-month treatment period, and then after a 6-week post-treatment washout. In each patient, we saw a significant improvement in the scale for assessment and rating of ataxia, as well as 8-meter walk test and 9-hole peg test after 1 month. And so we're seeing a rapid improvement of symptoms. Now, some of the patients were able to remain on the observational therapy for a longer period of time. In the video you see here playing on the right is actually a patient who's on treatment for 11 months. At baseline, he was completely wheelchair bound, unable to stand even with strong aid. But after 11 months, we're seeing a significant improvement in his functional ability. So again, these have also been published and is part of the comprehensive data set we have supporting the use of this drug for AT. Next slide, please. So on the basis of that observational and preclinical data, we set out to formally investigate IB-1001 in a multinational clinical trial for AT. We designed a master protocol that could investigate IB-1001 both for AT, but also Niemann-Pick disease type C and GM2 gangliosidosis. Because the drug seems to have both symptomatic and long-term effects, we distinguish the study into 2 different phases. The parent study, where patients are observed during a baseline period, a 6-week treatment period, and a 6-week washout period, and then an extension phase where they're looked at 2 1-year open-label treatment periods. We are enrolling patients aged 6 years and older into this trial. There is no cutoff for the upper age limit. Next slide, please. So we have completed the first 2 studies with the exact same protocol, same drug for Niemann-Pick disease, type C, and GM2 gangliosidosis. So these are the results from the MPC study, and we were very pleased that the trial met both its primary and secondary endpoints. We saw statistically significant and clinically meaningful symptoms, functioning, and quality of life. The boxes you see in blue here are the changes that occurred during the treatment period. So there was the improvement while patients are on drug. And the boxes you see in orange is the deterioration that occurred when patients stopped taking the treatment. So that's comparing the last day patients were on drug to 6 weeks after stop taking treatment. And so we see a nice bell curve, improvement during the treatment period, return to baseline during the washout period. And the consistency across the primary and secondary endpoints really reinforced the treatment effect. Next slide. What you see here are the results from the second trial in GM2 gangliosidosis. Again, we were very pleased to share that the trial met its primary and secondary endpoints and had almost identical results to MPC in regards to statistically significant and clinically meaningful effect. Next slide, please. So the status of where we are with the trial for ataxia-telangiectasia, we're actively recruiting the study at 5 different trial sites in the US, UK, and Europe. We've screened over 2/3 of the eligible patients for the trial, and we're still actively screening. COVID-19, as you could imagine, did significantly delay and impact recruitment. So we had started recruitment in January 2020. We essentially lost almost all of 2020 to COVID. But as they've been able to, and as local restrictions have changed, the sites have done a phenomenal job of screening patients. So the details of each of those sites is on clinicaltrials.gov in case you're interested in this study. And last slide, please. And I've listed as well my contact details here if you have any questions, as well as the details for the clinicaltrial.gov web page that describes the full protocol for AT, as well as the details of the principal investigators in each site. So thank you.
Taylor, that's really, really useful. Please don't go away because we definitely have had questions come through while you've been talking. Mark, I'm going to let you start off though.
Yeah, thank you very much. And thank you for that, Taylor. And you mentioned in your presentation around this sort of difference between what you're researching here and L-leucine, which is a supplement that is available in the marketplace at the moment. I'm very aware that we have many people in the audience who are non-scientists, non-clinicians, and actually are themselves patients or carers. So is it worth us being really clear here now that that is different? And also it could be mistaken for another supplement, N-acetylcysteine. I mean, it sounds very similar when said very quickly. So we can be really clear that this is what you're talking about here is not currently available, and that people shouldn't go out and buy those things and dose themselves with it expecting to see your results. And is that the right thing to be saying?
So the best way to describe it is, well, first, correct, absolutely. There will be no clinical benefit with L-leucine. The way that the body takes up L-leucine is the best way to think about it as a revolving door. So you could have a line of 100 people or 100 L-leucine molecules. The amino acid transporter can only pick one up at a time. So it doesn't matter if you take 100 kilograms of L-leucine. Because of the limitations of the transporter, only one's getting picked up and dropped off. When you acetylate L-leucine, so you do a chemical modification, which is the definition of creating a drug, it allows a new transporter to come, and it gets all those 100 people, puts them in a bus, and delivers it to the body. So it's not just that you can't take enough L-leucine, it's that chemical change, which needs to be done in a controlled GMP environment, that makes it into a drug and allows it to have that clinical benefit. So taking L-leucine will have no effect.
Also, I suppose, Taylor, as well, I mean, this is in very early stages, isn't it, this research? And we've had a question about... Sorry, say again.
I'll just clarify. We've completed now 3 late-stage multinational clinical trials. So we're entering the pivotal study for MPC, but pretty far along in regard to the development program. Apologies if that was not clear.
Fine, but then it will lead in clarify this question, because trials do take a long time. Someone anonymous has messaged saying trials take a long time, but what all families want to know is if the initial trial sees success, how long until this may be available to AT patients globally?
So our goal is that on the basis of the initial trial, as well as the pivotal study we're doing for another disease, at least in Europe, that will be supportive of a marketing authorization. In the US, the FDA has different requirements, and it still needs to be determined if we'll have to do another study. But in Europe, based on our conversations with regulators, provided we have successful placebo data from another trial we're running, we can use any successful secondary or phase 2 trials, to support the application. So a best case scenario, we're hoping that within the next 3 years in Europe, it could potentially be on the market, and we'll need to determine with the FDA if that's a possibility.
So, okay, bearing that in mind, I meant to say the trials are obviously in early phases, not the research. But so can any family, someone is asking, seek this treatment?
The only way you can access this treatment is by participating in the clinical trial. So if you're located in the US and Europe or the UK and you fit their inclusion criteria, then you would be eligible to be screened. But otherwise, it's still an investigational product, and it's only accessible in these official clinical trials.
So thank you, Taylor. And where can people find out if and how they can enroll in that clinical trial?
Sure. So the best is to visit clinicaltrials.gov. And if you search ataxia telangiectasia or N-acetyl-L-leucine, all of the details for that trial will come up, including the inclusion criteria, the schedule of visits, what assessments are performed. It also lists each one of the clinical trial sites, their contact phone number and email, and it has my details as well. So if you email me directly, I can put you in direct touch with the site.
Taylor, another question. Do you expect functional improvement for all ages? What's the hypothesis at this stage?
So the hypothesis is that it gets a little complicated now. We have an observational study seen patients as young as 6 months and as old as 93. And we've seen consistent improvement across the age range. In our MPC and GM2 trial, we recruited patients 6 to 74, and the subgroup analysis showed all patients improved. So we do expect that. Now, we do not expect symptomatic improvement in every patient. This is where it gets really tricky with the mechanism. Our guess right now is it's probably 60 to 70% will experience symptomatic benefit. The mechanism for this long-term neuroprotective effect is different, and we expect the majority of patients will have some form of long-term treatment benefit. But obviously, that's difficult to quantify just given the variability in the disease progression, which is why we're looking specifically at these tangible symptomatic improvements.
Does that surprise you that across all age ranges you saw similar improvement?
It's consistent with the 10 years of observational data we have, and again, 20 different disorders. And the mechanism itself isn't, it's not targeting something that's specific to AT or MPC or specific to a disease. It's targeting high-level neuronal dysfunction. And so it makes sense then that because it's not targeting something that's specific to the disease course, but the consequences of the neuronal damage, that we'd see benefit across each demographic.
So we have another question from Margarita Doria. It's quite a popular question. Could other clinical features such as immunodeficiency also be improved through this approach?
Possibly. Possibly is my only answer. We haven't obviously conducted any formal clinical studies in that regard, or even preclinical studies, so I don't want to comment, but possibly, and it's something that we should look into.
One more, and you're not going to be surprised, but it's regarding enrolment. You were giving some details of how to. Is there going to be a limit as to how many people can enroll?
So we're targeting 30 patients for the trial. We've enrolled 15 patients already, so we're about 50% through recruitment. But I would say that if patients are interested in the next few months, it will absolutely be a possibility. We've had a lot of challenges with COVID, so we're very hopeful that things, recruitment can pick up as the world hopefully returns to a little bit more of normality.
And we certainly are hoping for that, aren't we, in the world? Taylor, thank you so much. Taylor Fields there, Chief Product Development Officer and Senior Vice President of Intra Bio Limited. Thank you, Taylor. So that concludes our research presentations today. I hope you enjoyed the webinar and of course, with the help of Mark, have a greater understanding of some of the exciting projects which are being funded and supported by members of the Global AT Alliance. Recordings of all the presentations will be available in the next few days. Sean Kelly is going to be in touch to provide download links and request your feedback about today's event. In the meantime, you can visit the cureat.org website for details of various research studies and clinical trials. The content has been translated into Spanish, Italian, Turkish and French. And if you click on the 3 lines in the top left corner of Slido, you'll see a link which will take you directly to the cureat.org site. And finally, before we leave you, Dr. Mark Toms, always a pleasure and always an education. So thank you very, very much for joining me and all the speakers, of course, who have taken time out of their busy schedules to be with us today. I hope you found the webinar useful and informative, and we're going to use the poll button just one more time to provide your overall feedback. Thank you for supporting us and do take care.